Welcome to the deep dive. Today we're looking at how cancer genomes get so messed up, specifically those huge chromosomal errors. And there's a really surprising finding right off the bat.
Yeah, it's about the rate these errors happen spontaneously.
Exactly. We know cancer cells are full of these chromosomal abnormalities, CAs, but this paper measures the actual rate they pop up. And the big surprise, just losing one gene, the tumor suppressor TP53.
Which is super common in cancer.
Right, just losing that doubles the rate these errors form. And maybe even weirder, the basic way these errors happen seems biased towards losing chromosomes, not gaining them, even before cancer really gets going.
It really tackles a huge question. How do these massive chromosome rearrangements actually begin? We've known for a while they come from errors during cell division, mitosis. Like a chromosome gets left behind and forms its own little bubble, a micronucleus. But connecting that initial screw-up in a living cell to the final DNA sequence change, that's been, well, tricky. Genomics just gives you the endpoint, the snapshot. And watching live cells is just too slow to catch these rare events and see what happens next at the DNA level.
Right, the classic chicken and egg problem. So they needed a new tool. Enter MAGIC. Sounds impressive.
It is. MAGIC stands for Machine Learning-Assisted Genomics and Imaging Convergence.
Okay, break that down. How does MAGIC actually bridge that gap between watching the cell mess up and reading this resulting DNA error?
So it's this really clever closed-loop system. First, it uses adaptive imaging, think super fast microscopy guided by machine learning. It scans thousands of cells looking for any with weird nuclei, like those micronuclei forming, things that suggest trouble.
Finds the cells in crisis mode.
Exactly.
Yeah.
And the moment the AI spots one, bam, it photolabels it. It uses a protein called BENDRA2 that changes color from green to red when hit with a specific light. So it literally tags the cell while the error is happening.
Okay, so you've marked the suspect cell, but these are rare events, right? How do you sequence just that one cell?
That's the next step. They use FACS fluorescence-activated cell sorting to physically pull out only those red-tagged cells. And then, crucially, they use a special sequencing method called StrandSeq.
StrandSeq.
Yeah, it keeps track of which chromosome copy came from which parent the haplotype.
Okay.
So you can see exactly how the maternal or paternal chromosome got rearranged. The whole MAGIC system makes it possible to finally link that live error to the specific DNA damage and do it at scale.
All right, so they have the platform. What happened when they looked at normal, non-cancerous cells? What did that baseline spontaneous error landscape look like?
Well, first they confirmed something suspected. Dicentric chromosomes are key drivers. That's when one chromosome accidentally gets two centromeres.
The books that get pulled during division?
Right, so it gets pulled two ways at once, snaps, and causes chaos. But the really striking thing was the bias. It turns out the cell's own error process is inherently destructive.
Destructive how?
In these normal cells, they found way more chromosome losses than gains. Like 19 whole chromosomes just vanished spontaneously, compared to only 2 instances where a whole chromosome was gained. Nearly 10-to-1 loss versus gain.
Wow, 10-to-1, and that's before any kind of selection pressure from cancer development.
Exactly. It suggests the basic mechanics of mitotic errors lean towards losing genetic material.
Okay, that's a fundamental bias right there. So now the big question. We know TP53 is like the cell's emergency brake, stopping damaged cells from dividing. What happens to the CA formation rate when you cut that brake line?
Yeah, this is where it gets really relevant for cancer. When they looked at cells engineered to lack TP53, the number of these de novo spontaneous CAs shot up.
How much?
The overall rate pretty much doubled. In the cell line they studied, MCF10A, it went from about 13% of cells having a new CA after division to over 30%.
A jump from 13% to 30%. That's huge. So is TP53 loss changing how the chromosomes break the type of damage, or is it just that the errors happen more often because the checkpoint is gone?
That's the key insight. It's almost entirely about frequency. The types of errors and the way they resolve into rearrangements, pretty similar to the normal cells, but without the TP53 checkpoint saying, hold up, something's wrong.
The cell just plows ahead.
Exactly. It just keeps dividing despite the flaws. So you get way more initiating errors like those dicentric chromosomes forming bridges that snap. The downstream mechanics are similar, but the frequency of starting down that path skyrockets.
Okay, so the break prevents the start of many errors. And this MAGIC system could also tell them something about where the break happens, right? Location matters.
Precisely. This was another cool finding. They could induce breaks at specific spots using CRISPR. And they found if the break happened near the centromere subcentromeric.
Close to that central anchor point.
Yes. That specific location strongly favored the formation of things like isochromosomes.
And an isochromosome is basically a mirror image chromosome, like two identical arms. We see those a lot in cancer.
Exactly. And the thinking is, a break there leads to a structure that's maybe easier for the cell to stabilize and keep, whereas breaks elsewhere, like near the ends, often led to really unstable fragment isocentrics that probably just get lost.
So it's not just random breakage. The context of the initial break influences what kind of stable, potentially dangerous rearrangement survives. Early tumor evolution isn't completely random,
That seems to be the implication. It means the location itself provides a sort of early filter.
which makes magic not just a measurement tool, but potentially predictive. You could use this to test drugs, maybe, see if they stabilize things or boost those checkpoints.
For sure, it gives you a way to actually quantify if a drug is reducing the rate of these initiating events.
All right, let's wrap this up. What are the absolute key takeaways for our listeners?
Okay, three main things. One, the MAGIC platform works. It successfully connects the dots between seeing a single cell mess up during division and the specific complex DNA rearrangement that results. It closes that loop.
Got it. Link established.
Two, losing TP53 is bad news for genome stability, basically doubling the baseline rate of chromosomal abnormalities. And it does this mainly by letting more initial errors slip through, increasing the frequency.
Checkpoint failure means more errors get started.
And three, where a DNA break happens physically on the chromosome really matters. Breaks near the centromere are more likely to form specific structures, like isochromosomes, that we often see surviving in cancer. Location influences outcome.
Excellent summary. And here's a final thought to chew on. That huge bias we talked about, the 10-to-1 preference for chromosome loss over gain, happens right away, before selection. So the fate of a chromosome that gets lost in mitosis, maybe ending up in a micronucleus, seems decided very early. What are the specific cellular processes maybe related to DNA replication problems inside that micronucleus or pathways that actively eliminate it that make chromosomes so much more likely to be lost entirely rather than accidentally gained?
That's the next frontier, right? Understanding those very early decisions.
Exactly. What dictates loss versus gain right at the source? That's what platforms like Magic will help us unravel next. Thanks for the deep dive.
My pleasure.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.